Okay, we're going to be going over the mechanisms of pathogenicity, which is chapter 15 in the OpenStax book in this particular video. Again, just little parts of it. First thing we're going to talk about here is signs versus symptoms. Whenever a person has a disease, there of course have to be something that helps them notice that they have a disease. Signs, when it says that they are objective and measurable, these are things that doctors, nurses, EMTs, medics, whoever, they are going to be recording these things in the chart. These things tend to have a little bit more importance as they are recorded because they are things that were measured with instruments. So temperatures, thermometers, blood pressures, sphygmomanometers, EKGs, any device that can give you a specific measurement, that is something that's a little bit more reliable in terms of giving us more information. From a bacterial perspective, that's us taking samples from the person, growing those, and getting cultures from the person so we know exactly what organism is in that person. Now, symptoms, those are more subjective because they are being reported to us from the patient. They are still very real. It's just that unfortunately, from a medical perspective, they hold a little less weight because they are being reported to us from the patient. So that's the patient telling us it hurts. Well, we can't measure how much it hurts to us, and some patients are going to lie to us. That's the patient telling us that something doesn't feel good, but maybe not being able to elaborate on what that means. And so we don't know what it doesn't feel good means to them. And so symptoms are a little bit less reliable when it comes to information that we're going to be putting in their chart. And so we want to use those devices to get as many signs as we can because, again, it's just more reliable information. The picture, again, if you scroll over the pictures, when you have the PowerPoint, it'll tell you what that is, but this is a person who has necrotizing fasciitis. She has a bacterial infection, and the bacteria is producing a toxin that is essentially dissolving her connective tissue, so her skin is falling off. We can see that on her. That is a sign that we can write in her chart. She's going to report that it hurts, but we can also feel she's got a temperature going on. We're going to see that she has a drop in her blood pressure, and she's probably going to feel shocky. That is all stuff that signs and symptoms are coming together and telling us she's got a major problem there. Asymptomatic versus subclinical infection. The only thing that I want to mention here is this video. If you've never heard of Typhoid Mary, I really recommend that you watch the video on Typhoid Mary. She's a very interesting person. Typhoid Mary is sort of one of the first people that showed us that sometimes Koch's postulates can't actually give us all the information because she was colonized by the bacteria that causes typhoid, and she was spreading it to other people all willy-nilly all over the place, and yet she was completely asymptomatic, which is why she did not believe she was the one that was causing the infections and I believe killing some people. I guess I need to rewatch the videos to remind myself, but I'm pretty sure she killed some people. I know she attacked some people with some serving forks. She was a very interesting person, but subclinical infections means that they aren't showing those signs and symptoms, but they could still be contagious and spreading that infection. Okay, now your book has this chart on the different periods of disease or phases of disease. This is for both an acute, well, really for an acute infection and the first part of a latent infection, although then it comes back later. So incubation, what I need you to understand is it's different for every illness. For example, the incubation period for chickenpox is 10 to 21 days. So you get exposed and you might not show symptoms for up to 3 weeks after you have been exposed to chickenpox. Then you're contagious 1 to 2 days before you get the rash. And so you were spreading that infection before you even realized that you had a problem. Then you continue to be contagious as you have the itchy rash and the fever and the horribleness, usually for about 7 to 10 days until the blisters start to scab over. You've stayed contagious that whole period. The incubation period for the flu, on the other hand, instead of being nearly 3 weeks, is only 1 to 4 days. So it has a much shorter incubation period. It's only contagious for 1 day before symptoms start, and it's contagious for about 7 days after the symptoms end up resolving. So it's different from what you have for the chickenpox. So again, everything is different in terms of how long the incubation period is going to be, when you may be contagious. all of that stuff varies illness to illness, but they're going to tend to go through these phases. It's just how long it takes you to go through those phases is going to vary. Also, it varies depending on the health of the person. A person who's immunocompromised, they may be contagious for a longer period of time than a person who is not immunocompromised. Okay. This picture I had pulled from a different book and I just liked it, so I wanted to share it with you. So acute goes through all those phases from just a second ago. The incubation period, the prodromal period or phase, which they didn't show here, illness, period of decline, and then the convalescence period. Then it's over. It's just gone after that. So that would be like the flu is a good acute infection. Chronic, on the other hand, it has the incubation, the prodromal phase, and then you are just ill with that and it doesn't go away. Hepatitis, like hepatitis B, hepatitis C, those are good examples of your chronic infections, although we do have treatments for hepatitis C at this point. Hep B, not as many for that one. Latent infections, the late great comic George Carlin called those the gift that keeps on giving, because you get them and then they come back around. So you go through the incubation period, the prodromal phase, illness, then convalescence, and you feel like everything's better. But then after that period of, yay, I'm all better, it comes back around and you get another period of prodromal phase, then illness, and then convalescence. And it can do that over and over and over again, dozens, if not hundreds of times, depending on how long you live, what the illness happened to be, lots of different things. Herpes is a good example of that one. One thing that still a lot of people don't understand is chickenpox is a latent infection, because chickenpox, you run through that first round of chickenpox and then it hides in your nervous system and then it comes back as shingles later on. So it comes back as something slightly different, but it's still the varicella zoster virus that causes both of those infections. Okay, Koch's postulates are important even though they don't always get you to the right answer. One of the reasons that they are so critically important is that they help prove the germ theory of disease, that certain specific microorganisms caused certain specific diseases. In his case, Bacillus anthracis caused anthrax. He did some other ones as well, but anthrax was one of the ones that he is better known for. You can see his postulates written out here and in picture form. I've given you some of the pros and cons here that kind of go along with that, but some of the cons that go along with some of his things, there are a lot of diseases that we can't test in mice because they don't cause disease in mice. They only cause disease in humans. And it is completely unethical to just isolate a disease from a person and inject that into a healthy human to see if it causes disease in a healthy human. Not to say we haven't done that before, we absolutely have, but we're trying to be better people at this point, and so we don't do that in the United States anymore without informed consent and a whole bunch of stuff. So yeah, Koch's postulates really hard to do in human cases in a lot of cases. He also already mentioned he didn't take into account that some people could have asymptomatic infections like typhoid Mary from earlier. He didn't take into account that not all organisms could be grown in pure culture, like viruses, prions, new pathogens that he didn't know about back in his day. So nowadays we've taken it a step farther and we have molecular Koch's postulates, which is sometimes there's different strains of bacteria, and the strain is what causes disease because of a specific gene that that strain has that allows them to produce a specific virulence factor, like a toxin or an exoenzyme or an adhesin or something along those lines. And so those genes give them a phenotype that allows them to be more pathogenic. That gene, we can genotype for that. It allows for PCR and Western blots and just more genetic information. We're not relying on organisms in a lab as much as we used to back in the day. Okay, the stages of pathogenesis. If an organism wants to cause a disease in you, it has to be able to do all of these things. First, you have to be exposed to it. Like there's a virus out there that I'm sure could wipe out humanity, but we haven't met it yet. When we do meet it, we're going to be in trouble. But because we haven't been exposed to it yet so far, we're doing okay, although COVID did do a number on us. After we've been exposed to it, that pathogen has to be able to adhere to the correct cells in our body that it needs to be able to infect. So that adhesion or colonization is going to be important. That is genetically determined. Once it sticks to our cells, it needs to be able to invade those cells and potentially invade deeper tissues to get into the body and then infect that body and cause symptoms so that it can spread to others from there. Okay. Couple of concepts that I wanted to elaborate on is portal of entry and then portal of exit. This is how do things get into you versus how do things get out of you. This relates to how do you actually contract your illness and how do you spread your illness. So some things you can get through direct contact, which means you touch somebody and you get it. That would be like HPV, human papillomavirus. That is a direct contact. You touch somebody's skin. So the portal of entry there is skin. Pink eye, that's an eye thing, which it's part of the respiratory system in a weird way, although some books do put it in the nervous system, but whatever. Pink eye, you get that through the eye, so its portal of entry is actually the eye. Depending on how you get an ear infection, if it's otitis externa, you get that through the ear. If it came in to the middle ear, it probably came in through the throat, so that's through the mouth or the respiratory system. If a baby gets it while still inside mom, its portal of entry was the placenta. So this is just kind of showing you how things can get into your body. One thing I always like to mention here is very, very few things can actually infect you through your skin unless your skin is broken, aka you have a wound. So the issue here is we do tend to have wounds all the time. We have a bug bite, or we've scratched ourselves, or we cut ourselves shaving. But if your skin is unbroken, most things aren't going to be able to get in that way. Mucous membranes, on the other hand, those are not as sturdy as your cutaneous membrane. And so notice that a lot more of this stuff is mucous membrane. Okay. Portal of exit, this is like the slide says, how things get out of your body. A lot of this is going to be through secretions or body fluids, but your respiratory tract, just breathing, sneezing, you can release a lot of your respiratory infections that way. UTIs can come from the digestive system or from the reproductive urinary system. Every specific pathogen has its own specific portal of exit. That leads to how it can end up infecting the person. Okay, adhesins and tropism. Now here, this was the book talking about viruses specifically. So viral adhesins are how viruses attach to your cells. And again, if they can't attach, they can't infect you. So those adhesins, since we've had COVID now, hopefully you guys have all heard of spike protein. Spike protein is the protein they use to attach to the ACE receptor that's present in a lot of our cells. Once spike attaches to ACE, the COVID virus gets into our cells and causes the actual infection to take place. It takes over the cellular machinery, makes your cells little COVID factories at that point. So those adhesins, they have what's known as tropism, which means they can only bind to cells that have that ACE receptor present. If the cell doesn't have the ACE receptor, they can't infect that cell. This is one way that they can get to the cells they want to infect, which unfortunately a lot of the cells of your body have that ACE receptor. So that's why COVID can do so much damage to the human body. Also, a lot of mammals have the ACE receptor, which is why COVID could infect dogs and cats and mink and humans and pangolins and so many different species. But a lot of species have a more specific receptor and adhesin combination. An example that I gave you up here, parvovirus, is a virus that can infect dogs, but it can't infect people, where COVID could infect both dogs and people. So sometimes those adhesins, they have tropism that is more narrow, whereas sometimes it is more broad. The viruses that have a broader tropism, they're going to tend to be more successful and be more virulent out there in the world. To be more human specific, hepatitis B, it can infect liver cells but not muscle cells. And so it's got a more limited range within the human body because its receptor is only present in the liver. It's not present on as many cells as that ACE receptor is for the COVID virus. Viruses have the ability to change over time. It is why you are supposed to get flu vaccines every year. It's because the flu virus evolves, and so the vaccine that you got last year won't work on the flu virus that is circulating around this year. The flu does both antigenic shift and drift, but the shift allows flu to change more quickly. So drift is natural mutations that just accumulate over time. It tends to occur more slowly and just change the virus a little bit at a time. The problem with flu is that the flu virus has 8 different strands of RNA present within the capsid. If you get infected by 2 flu viruses at the same time, virus A and virus B, like it's showing you here, what happens is as the cell packages these up, it mixes up the segments of RNA that's present in these viruses, and it makes a new virus that didn't exist before. So right now, we have this problem with bird flu that has wiped out birds literally worldwide. That bird flu has not been that big a problem in humans at this point, and right now it is July 3rd, 2024. However, that bird flu has jumped into Texas dairy herds and dairy herds in other states in the United States. Epidemiologists are very concerned about that because once we know that it has jumped from birds into cows, and cows can get flu from multiple different things as well, we know this antigenic shift is possible. We know that those genomes are getting mixed up, and we know cows are in close contact with people. We know that there is a much bigger chance that the correct combination of things is going to happen to turn that bird flu into something that can then jump into people and start making people sick. Hasn't happened just yet, but it could be on the horizon because of antigenic shift. And so there are people who are very worried about it. And unfortunately, it is not as big a news as it really should be. So I don't know, there's your worry for this year and 2024. I don't know when you're watching this video, but hopefully it doesn't turn into anything, but it is a concern at this point. I gave you 2 different slides to elaborate on those a little bit more. One other thing that I did want to end with, this isn't really discussed in the OpenStax textbook, but most of the time, pathogens do not want to kill you, because if they kill you, they have just destroyed their habitat. They now have no place to live. The example here, if you did not know, rabbits are an invasive species in Australia, and apparently in Australia they hate rabbits very, very much, and so they built what was supposed to be a rabbit-proof fence. Didn't work. They've done a lot of different things. They've poisoned them. They've really done a lot of different things, and one of the things that they did was they released a myoma virus that used to be so virulent that it would destroy any rabbit that it happened to meet. But because viruses do not want to destroy their hosts, what happened is something called balanced pathogenicity, which is the virus over time became weaker and the hosts became less susceptible to the virus, aka the rabbits became less susceptible, so that eventually the myoma virus, it would make the rabbits sick, but it would not kill them. So that failed as well. The rabbit fence failed, the poisons failed, and the virus failed because viruses don't want to kill you. They want to breed in you and release themselves over the world. Now, that is anthropomorphic. They aren't thinking about this. They are not conscious, but their job sort of is to spread their genetic information, and they cannot do that if you're dead. And so balanced pathogenicity means they want to breed in you and they cannot do that if your immune system wipes them out. So they evolve to try to evade your immune system. Your immune system evolves to try to kill them. And it's this constant back and forth between your immune system and the pathogen. And most of the time, it works out so that you don't die. You get sick, but you don't die. But sometimes something happens and the pathogen got too strong, or you wipe something out. Hopefully we end up winning like we did with smallpox. But balanced pathogenicity tries to mean the virus is just strong enough to make you sick and breed, but not so strong that it kills you. Okay, that is the mechanisms of pathogenicity. There's a lot more in this chapter about the toxins and the exoenzymes and a whole bunch of other stuff, but I didn't think that those were too difficult to understand, so I just wanted to hit you with some of these things that weren't necessarily discussed or might need a little bit of elaboration. If you do have anything that you have questions about, please don't hesitate to contact me.